You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I've got a great guest. His name is Daniel Fels, F-E-L-S. He's been working in science for many years. He's currently developing his own laboratory and his own independent research. His laboratory is going to be called the Biophysical Cell Communication Lab. It's in progress, but doesn't stop him from doing his own research. He's authored a number of publications that talk about an additional form of cell-to-cell communication, where cells appear to use light to communicate. So we'll get into that. And Daniel, thank you for coming. How you doing?
I'm fine, thank you. It's a pleasure talking with you about my topic. Hello, Richard.
Yeah. So tell me, how did you first imagine or observe or think that cells could communicate using light?
You need to know that actually I was reading books and I came across such stories very early where I still didn't know what it actually means. But it all began in 1999 when a friend told me that DNA is emitting laser light. That sounded to me very crazy, but I took it serious and I began to look for the people that do this kind of research. And while doing this, I came across of papers, publications published in the 20s, 30s, 40s of the last century that cells are able to communicate electromagnetically with each other. This is actually how it started. And this was awakening my interest in such a profound way that I wanted to do my own experiments.
So the fact or the idea that cells, the DNA of cells emit laser light, again, you thought that was crazy, which sounds understandably crazy. But you were fascinated by the idea that cells maybe could communicate using light?
Exactly. You know, that DNA emits laser light. I wouldn't underline this today, but I think I do not think, it's just a fact, an observational fact that cells do emit light in the visible range, but not only in the visible range, also microwave or infrared and even radio waves. And my interest was less in what are the sources in the cells that allow the emission of light. And also my interest was less on what are the receiving parts in a cell. My interest was whether they can really use electromagnetic waves to signal to each other. And in order to find out more about this, one has simply to separate cell populations with a glass barrier from each other. This disables chemical communication. And exactly this is how I began my experiments, separating cell populations with a glass barrier.
How come you didn't want to look at, what was happening in the cell to generate the communication or to receive it or interpret it? Is it just too much of a task to try to do all of that? And the first thing needs to be proof that it actually even happens? Or what was your thinking?
Yeah, I mean, there are two answers to this. One answer is that you need a methodology to investigate such questions. And my education led me into another direction. During my studies, I became interested in behavioral biology. And a lot of animal behavior has to do with signaling and with communication. So you can say my brain was kind of focused on this type of questions, therefore less of interest in the source and receiving part, but a primary interest whether this phenomenon occurs at all, first of all, you know.
So what does your experimental setup look like? And, then we'll get into some of the observations.
Yeah, it's actually a very simple setup. I'm using little vials. We can call it little aquariums. They have 2.5 centimeter by 2.5, and they are 4 centimeters in height. And it's like a little glass tank. And then I have even smaller glass tank, which I can neatly place into the bigger one. So you can imagine that you have a little tank that stands in a bigger tank, but all of them are very small. And in each of them, I have a medium, liquid, water, with some feeding food for the cells I'm working with. And I do have this in both vials. And as a control, I have the same setup, but in one vial there's only medium, but no cells. And this other setup serves as a control. Was that clear enough?
Yeah, so you have these two little, I'll just call them little fish tanks or aquariums. And what are you putting in them? What kind of, bacteria or creatures are going in?
Yes. My study organism, its Latin name is Paramecium caudatum. It is quite a huge cell. It's an organism that lives unicellular. So it's one cell, that's the whole organism. And they have 0.2 to 0.3 millimeter in length, which is actually huge for cells. And therefore, it's very simple to count the number of these cells under a binocular. So I'm working with them. It's a famous organism used worldwide for different types of studies. And when they are in a tank, there are also some bacteria there. They feed on bacteria. And when they have fed enough, they have enough material in their body to double them, to make a so-called cell division. And it's actually, this is my indicator for effect. Whether having neighbors or not has an influence on cell division rate. Because the very early studies in 1923 indicated that cells can have an influence on cell division rates in another group of cells, but the signal is not chemically transmitted, most probably. It was at that time called a radiation. Today we call it an electromagnetic wave.
So how can you observe the paramecium? Can you use a camera setup with, 2 or 4 times magnification, or do you need a microscope to see them?
Yeah. Actually, it's very simple. I put the whole setup is during the period of an experiment in a black box, in a literal black box. So I keep them all, these pairs of aquariums, as you called them before, I keep them in a black box for 2 days, for 48 hours. Before I place them, I know how many cells are in each tank. And then when I take them out, I count them. And then with that, counting them under a binocular, I can deduce what the effects of having had a neighbor were.
Why not set it up where you have a camera or 2 cameras that watch each of the aquariums from the side? Then see them interact.
No, I do not have this, and you wouldn't see anything. You wouldn't see anything with a camera. And what I realized, I have to tell you this is important, these cells would not show effects on each other if there were light. I cannot really explain why this is so, but under light conditions, I do not find these effects. So the phenomenon seems to be a kind of a very weak phenomenon, but occurs under the conditions of my experiment. And we have to be aware that we are doing here basic research. There's not so much we know about electromagnetic cell communication.
So you can't directly observe
No,
the electromagnetic being
no,
passed between the cells. So what
no.
happens? You count the cells, you put them in pitch darkness for 48 hours, you
Yes.
pull them out, and then you count them again.
Yeah, I count them again. I can give you an example. I have in the outer tank, let's say I have 100 cells, and in the inner tank I have 5 cells. Then the two are together for 2 days in darkness, and at the end of that period, I take them all out and count how many they are. I have controls. In these controls, I have 100 cells outside and 0 inside, or 5 cells inside and 0 outside. And this allows me then statistically to find out whether having had a neighbor or not would affect cell division rate. And it does. I did several experiments where I could show that neighbors affect growth either by inducing growth or hindering growth slightly. And other effects were that the cells were eating more or they were eating less. These were the first results which I could publish in PLOS in 2009. And these were the results that made me sure that I'm on a way to discover a little bit more about electromagnetic cell communication.
So, yeah, what happened specifically when there was the 100 on the outside and 5 on the inside tank? What did you observe after 2 days, for instance?
Yeah, it depended on the material I was using. But let me, before I tell you what the results were, I would like you to know that I had not a particular expectation about the effect. I had only one question: is there an effect, and will I find it? That was all. So it was a little bit peculiar. When there was a quartz separation, the 100 cells had a reducing effect on cell division rates on the 5 cells. And when there was a glass barrier, then it was the other way round. The 5 cells inside had an inducing effect on cell growth in the hundreds outside. I did not really know how to interpret these data, but I would like to tell you something about the 2 different glass barriers I was using. Because I used 2 different glass barriers, and one of them being a filter for UV waves, and the literature was telling that the effective range is UV, I thought that glass would be a filter, not meaning that there would be no effects when I have a glass separation. And the first surprise was there was an effect, and the next surprise was that the effects were just differently when I had glass or quartz separation. But still, I didn't know what that means. Then I was looking at all results, and I thought quartz separation is closer to the natural conditions than glass separation. Why? Because quartz allows all electromagnetic waves to be transmitted from one side to the other. And this is closest to the natural situation when these cells are together, wherever they are. There is no glass barrier between them. So I was looking at quartz separation, and then I saw that with quartz separation, there was a pattern. There was always the same pattern. The bigger population had a reducing effect in cell division rates on the smaller population. So that was a pattern. And with that pattern, I could formulate now a first question. The question was not anymore, is there any effect from one population on the other? But the question was now, has it to do with the regulation of the density of these cells? Then I made an experiment where I tested exactly for that. And these results I could also publish in scientific reports. And I'm quite happy because it's a nice place to publish in scientific reports. And it's a nice result because there was an indication that the more cells there are, the bigger is the reducing effect on cell division in a neighbor tester population. And why is this interesting? Yes?
So for a given volume, you tested more and more cells, thereby making it more dense in between each cell. And the more
Yes.
dense it was for the outer population, you're
Yes.
thinking that that signaled the inside population, which was at constant density for each experiment, to grow slower because the outside
Exactly.
population was telling the inside, it's too cramped in here, don't grow.
I agree. No, wait, yes, what you said is perfectly right, but you are already slightly interpreting it, and I would like to step a little bit back and say this. Um, the dense population, and this is what I assume, is electromagnetically regulating its own density. Whether they know that there are a few cells in the inner tank or not, I have no idea. But if the bigger population, the outside population, communicates electromagnetically whatever about density, the signals, of course, go into the tank with the smaller population and they receive the signal, make less of cell divisions. This is how I interpret it. I'm not saying the bigger population is telling the smaller one grow less. The bigger is telling within itself, we are many, we have to stop growing, and this is electromagnetically organized. That's my interpretation of the results.
I see what you mean. Okay. And how much slower have you seen the inner population grow, or how much faster, for instance?
Oh, the exact numbers are now not in my head, but it might be that, on the high density condition, they were still growing, but on the low density condition in the neighborhood, there were many, maybe twice as many as in the other case. So it's not a tremendous effect, but it is an effect. And it was the first paper actually that gave an indication for an electromagnetic population density regulation. Terrible title, but that's what it is.
Did you reverse the situation and have a lot of cells in the inner chamber and a few in the outside and see if you had the same effect?
Yeah, in that case, I didn't do it. One can do that, but I just stick to that design. It's a good idea. One can do that, and as I'm working alone, it's quite an extensive work to make such experiments, many repetitions, and so I I'm less interested whether in an inverse situation the effect occurs again. I'm quite sure it would. If I have one day a group of people that does this work, then we can do all kinds of testings in this direction. First of all, I was only interested whether I'm thinking in the way the effect, or whether I'm thinking in the correct way that allows me to understand what the function of this electromagnetic signaling is. And with that experiment, I was happy enough that I didn't felt a need to do it in a different way.
Because I could see, instead of having the two tanks concentrically around each other, what if they were just next to each other? They just share a wall. And you have A and B next to each other. Would that change the dynamic of it? And in A, there's 100 cells, and in B, there's 5 cells.
Yeah. I still have to do this. The beauty about this is that the surrounding of the inner tank by the outer tank is 100% surrounding. So they are really in the midst of whatever communication is done by the outer population. When I have them side by side, you can say that there is a 75% of loss of light that goes into the other directions than the two walls that are attached to each other. But in the future, I will do that because 100 cells can have different effects on neighboring cells depending on the number of the neighboring cells. I obtained results where 100 cells would push cell division if there were only one cell placed in the inner tank. That one cell grew much better than a single cell without neighbors. While when there are already a few cells in the inner tank and there are 100 cells surrounding them, then the effect is opposed to that one. They reduce cell division rates. And my question is, in a future experiment where I have 3 tanks side by side by side, I would have in the middle 100 cells, to the left 1 cell, and to the right 5. And here my interest is whether these 100 cells, on the one hand, reduce cell growth in the 5 cells, and on the other hand, push cell division rates there where there is only 1 cell, or whether the 100 cells only go in a kind of interaction with one of the 2 neighbors. Each result would be interesting to learn more, but that's a future experiment. I haven't done that yet.
Well, what about from the less dense cells telling the more dense cells, go ahead and grow? Is there, I don't know, is there an authority that's created by the number of cells as well? Like, what if you had 50 in the inner and 100 on the outer?
Yeah,
maybe the 100 on the outer as compared to a control group would reproduce faster, because maybe the 50 on the inside are telling the outer grow faster, and the outer are telling the inner grow slower.
It's very interesting. I'm telling you, I love these questions. I love these thoughts. But unfortunately, yes, I'm an independent researcher, but my time, the time that is available to me is a little bit restricted. And it's an interesting thought, to go more into a dialogue, what they are doing. Again, it's time consuming. And if I had a group of researchers, then each one could make different experiments in that direction. My interest was how to continue, was on the one hand, whether this signal has a meaning only within a species or also across the species border. So I was taking different organisms in the outer and inner tank and just wanted to know whether they also have an effect on each other. So I took Rotatoria, which is a worm-like, very small organism, consists in about 1,000 cells and is only slightly bigger than a paramecium. And the results were just indicating that they can affect each other. The worms can affect the unicellular paramecium and vice versa. I was only interested whether there is an effect across the species border, because that's what some people claimed in the 30s and 40s, that there were such effects. And I didn't go on with that. I would like to stay within paramecia. And I had a result that was very astonishing. And this is where I will continue the experiments, that when there is mortality in a high dense population, so when they are dying during the 2 days, not totally, not down to 0, but let's say I place in the little tank 300 cells, and 2 days later I count 220. So about 80 were dying. That's what I mean by mortality. So when there was mortality, then there was not a reducing effect on the small inner population, but the opposite. And it was even correlated in a fascinating way. The more that had died in the big population during these 2 days, the more the 5 cells inside grew. And this is interesting. It is as if they perceive death on the other side. And I call that for myself death compensation, just to have a term. This is the kind of experiment I will pick up in the future to find out whether this is really so. Because I had not tested for it. It was a side observation of an experiment, and now I will test for that.
What about if you put 2 populations there and they're equal, but you give one food and you give one no food for a day? I mean, you can do that.
I could do that. It's interesting to think along these lines. It's not so easy, because when they have no food, they will die. And then you cannot say that those with cells have a detrimental effect. those 100 cells that got food had a detrimental effect on those that didn't get food, because not getting food is already a problem.
How long does it take for the paramecium to die if they don't have food, for instance?
Oh, it's no problem in a week and less.
Well, if you did it for like 2 days, food, no food, equal population numbers. I'm wondering if the ones that had no food would tell the other ones that had food don't grow, even though they had food.
Okay, I get the point. It's interesting. It's really interesting. Yeah, I like the idea. Could be. I cannot predict. See, I do not understand enough of the system to make big predictions. I'm just testing for the effect and publishing it so that scientists can take the phenomenon serious. This is not storytelling. There's really something going on. And cells are not just a package, a well-organized package of molecules, of chemicals. There is more going on in cells. There are electric beings as well. They have electrostatic and electromagnetic fields. And my motivation is just to make a few good experiments that nicely demonstrate that there is something non-chemical going on between cells, and that this is most probably an electromagnetic system of communication.
What about, you said when they're exposed to light, this doesn't happen? Like, what happens if you just have this setup sitting there in the lab on the counter, for 2 days? Do you observe anything?
No, I wouldn't observe. The effect of light is almost dramatically, it can quickly disturb the results. I was astounded by that, I had an experiment where I was looking at feeding rates, and in order to see how much they feed, you need to prepare the cells, which takes time. So you have to kind of kill them, fixate them, and then count them with a microscope. So each pair of cells that I took out of the black box, I needed about 20 minutes to get that all done, to get the numbers of how much they were eating. And always when I took a pair of aquariums, as you called them, when I took a pair out of the black box, there came a little bit of light into the black box. Not much, and only shortly. But the amazing thing was that the first pairs I've taken out all showed an effect. The last pairs, however, didn't show any more an effect. So the impact of external light is, I could say, dramatic with regard to my experiment. Yeah, it was almost immediate. And this brings us to a critical point, because these cells under natural conditions, I would say, never experience such a total darkness as in the conditions of the experiment. However, in a pitch dark night, deep on the ground of a pond, it might be as dark as under my experimental conditions. It might be. So this makes the whole experiment, on one hand, even more interesting, because if we allow us to think that these cells never experience the conditions they experience under my experimental situation, then why should there be such an organized result? It's not a stochastic result. It has a structure. And therefore,
I think that, if these things are hanging out in a pond at night, it sounds like a backup form of communication. At night, it would be dark, and maybe at night there's no predators, for instance, and then they can communicate this way and, worry about dividing. But in the daytime, they have to worry about not being eaten or eating themselves, and maybe that takes precedence.
Makes sense. Makes perfect sense. It might be a night communication. But even in a night, there are photons around from moonlight and stars. And I got the feeling from experiments I made that a little bit of light already can do it. But I agree with what you say. It makes sense, and it's quite possible that it's a night strategy. If we allow us to think that already a little bit of external light doesn't allow this communication, then we are looking at a system that cannot have evolved under conditions of what we call natural selection. And that makes it also interesting to me. I believe that the cell internally within a cell, that the organization of keeping the order upright within a cell is not just driven by chemicals that meet each other and ATP, the so-called battery of the cells, is delivering enough energy to do it. I believe that the electromagnetic fields play an organizing role in the whole life processes. And when it's dark enough, then the different neighboring cells kind of connect with their internal organizing principle among each other.
I guess, to me, it's an adaptation. It's another way to communicate. Again, it may be backup. It just may be in certain conditions. Mm-hmm. But you could also wonder, well, if a lot of the time they're exposed to, ambient light, why not communicate with light, but just a type of light that they don't normally get in ambient conditions, a different wavelength, so that they can communicate all the time, no matter the outside condition, the outside light condition.
Yes.
Why not do that? But,
it's really possible. What I understand about cells is that's also maybe something we should note here, is that cells cannot avoid to be electric. The majority of all their molecules have charged surfaces. So there's a lot of electrostatics going on. And second, when charged structures move rhythmically, let's say when they oscillate, an oscillating charged particle already emits an electromagnetic wave. And further, chemical reactions can either need energy, or the products contain less energy than the chemicals that reacted together with each other, leading to that product. And the energy difference is then an electromagnetic wave. So there are electromagnetic waves, and it's actually rather surprising if we think that the cells would not use this, or even stronger, would be able to avoid being affected by those electromagnetic fields that they generate during their internal processing.
That's true. The cell is metabolizing all the time. Like you said, its membranes are charging and polarizing, depolarizing.
Yes.
So it makes you wonder if another cell could recognize and read what's going on in its brother or sister cell, and recognize those pulsations and that rhythmic changing of potentials. And I don't know what that would tell it, but you probably can tell it, I guess, the degree of metabolism and a whole bunch of other things.
That's exactly what I believe. And the thing is that there's not yet a lot of attention on this phenomenon. In biology or in life sciences in general, there's a focus on chemicals. Of course they exist. Of course they are effective. I'm certainly denying that. I'm not stupid. When I stand on the weighing machine, I'm weighing the sum of all of my molecules. Of course they are, and they are effective. There are hormones, etc., etc. What I'm just saying is that in addition to this material that has weight, there is a weightless component, and that's the electric field. And this field plays a significant role in cell dynamics. And as you said, I fully agree, this can also be perceived by a neighboring cell. And then the step to understand that there could be a communication system that includes the use of electromagnetic wave, that's a very small step. And actually, I'm just trying to show that this really exists, that they really can connect across a glass barrier. I say again, very carefully, most probably using electromagnetic waves, but certainly not chemicals, because they cannot trespass the glass barrier.
Right. What about if two neighboring cells were able to feel the shape and the change of shape of the neighbor's electric fields? Maybe that's a way of communication without having to exchange any photons. I don't know.
I mean, what you are referring to is something that's not what I'm doing in my experiments, which doesn't
I know.
matter.
I'm just
Good,
speculating.
of course. And Richard Funk, for example, he published a paper where he can show that in embryology, when an embryo develops, that first of all, on the surface of the embryo, there are different charges, plus or minus charges on the one hand. And on the other hand, some of the cells in a developing embryo, they do not stay on the place where they appear due to a cell division, but they start migrating. And they could observe that the cells themselves are polar. So one part is plus charged and the other end of the cells is negatively charged. And then it's these charges on the surface of the embryo and on the surface of the migrating cells that attract each other. So the electric field or the electrostatic field seems to play a very significant role when embryos begin to organize themselves.
Right, and how would the cells know how to configure themselves in such a way? Where does that plan come from that says be polar in this way, positive here, negative here, and so that the cells can migrate to the right spot, the right orientation, and everything?
Yeah, the plan of all of it. I think that's the most. Here, I'm very, very, how to say, I'm very humble in saying anything. I'm humble in saying there is a plan. I'm humble in saying I know where the plan is. I'm humble in saying this cannot be the plan. I must confess that to me the whole developmental process is really enigmatic, which, however, doesn't mean that we do not know a lot about it. We know that if certain genes are not switched on, the process stops. That's okay, but that's not answering what is giving form to the organism. It's just saying that if the brick stones lack, you cannot construct a house. It doesn't. The lacking brick stone is not an explanation regarding the architecture of the house. And I only think that the electrostatic fields play a role in the organization, but the plan is difficult. Sometimes I have a feeling that there's just a rolling planning. It starts in a particular way, following certain rules. And if you just follow these rules, then there is an organism at the end. But this is not a scientific explanation. I fully agree. It's just giving proof to what I said in the beginning. I do not know how this is organized, to be honest.
So why is it? I mean, I love science. I don't have a lab and I'm not a scientist. Like, to me, it doesn't seem like a big deal. Like, why does it take so much time and effort for you to do more experiments? Like, what takes so much time and effort to do this? It seems like, I don't know, again, it's an ignorant comment, but why couldn't you do a whole bunch more experiments? What takes so much time?
Oh, because I'm earning my money as a teacher. I don't have so much extra time. And this is the major reason.
Okay.
I'm sometimes simply tired, and I'm doing it all alone. And over the last years, what I could do is and about this, I'm very happy. I could publish some results that of course haven't been published. So year by year, in 15, a book appeared that I edited together with two friends. In 16, I could publish a paper that showed effects across the species border. In 17, I could publish a paper on the population density effects. And in 18, I could publish an essay, which is called The Double Aspect of Life. And what I just see is that people are interested in it. And I hope that others start doing these experiments. I'm not paid, and So to answer your question, why I'm not making more experiments, they are time consuming because I have to make many repetitions. I need all my attention when I do them. And I just sometimes do not find the spare time anymore because there's too much other work I have to do.
Yeah.
And then I find the extra bit of energy I need. I went through a difficult period over the last years. This is also one reason why I didn't make so many experiments. I hope, again, here I repeat myself, that other people start doing these very, very simple experiments. And this could be the case. Allow me to add a little thing. There is an Asian publishing house that was asking me whether they could take my last article, The Double Aspect of Life, as a book chapter. And they can do that. There's the right. Now, the title of that book is The 5 Top Contributions in Biology 2019. So this group of people, I don't know them personally, okay? But this group of people obviously has the impression that what I'm here contributing is really important. And that's my wish and my desire, that other people recognize I do my very best to give good evidence that there is a phenomenon and to work with the fields of the cell. That's a field in biology that has been glaringly neglected over the last decades. And I think it deserves our shared attention. And by shared attention, I mean the attention of molecular biologists and bioelectrically interested biologists, and maybe also physicists and engineers to discover the electric body that we are at the end of the day.
Yeah. So what's been the reception from the papers you put out there and the research, the scientific community? Do most people say, sounds all right, and move on, or are they excited, or what kind of responses do you get?
The kind of responses I have is, for example, the 2009 paper in PLOS is now over 30,000 times clicked, or the double aspect of life has soon 3,000 clicks about 1.5 years after its publication. So it's the clicks that are an indication. If you compare the clicks with other papers, you can see that this is a little bit or a lot above average. The PLOS paper is about 3 to 4 times above average. Then I have a platform on ResearchGate, and on ResearchGate I can read how many people went on ResearchGate to download one of my papers. So I'm often visited there. That's another indicator. And then there are other indicators which I wouldn't. I do not give too much of weight, because once you are kind of in the radar, that's with a few good publications, then you get all the time invitations as a speaker to conferences. And also every week I have an invitation for a conference. Every week I receive an invitation to write an article in a journal. But to be honest, very often the topic of the conference has nothing to do with my topic. The topic of the paper the journals has nothing to do with my topic. And as there is today everything open access, it's business. It's business. When you publish an article today, you do it open access. And if it costs not so much, it's $600. If it costs a lot, it's $2,500. So it's a business system also for the journals. And of course, they are writing every day 1,000 emails around the planet.
Okay.
So that's not an indication. It's only an indication that I'm in the radar. But again, that people click and look at my works, this is evident, and gives me the feeling, together with all my other friends that work in a related way to what I'm doing or less related way, I have a feeling that there is no way to avoid in the future the fact that cells use their electrostatic and electrodynamic fields for the inner organization. There's no way to avoid this. If it takes 10 more years, okay. But this understanding will come, and in the near future, I'm absolutely convinced that we find these informations in textbooks, school textbooks, and student textbooks in life sciences, biology, and so on. There I'm convinced.
Yeah, a question occurred to me. How would you even know where in a gene or which, I'm just going to give an example. So if you assume that genes code for everything, where is the gene that codes for like the morphology of how an embryo grows? Where is the gene or genes that code for how to conduct cell processes to set up electric fields in a certain way and direction and intensity to create a communication system for the cell or to direct its own operations? Like, how would you even find such a thing?
Yeah, that's a fantastic question. I think that's one of the deepest questions we can ask to life. And the before evolutionary theory occurred, the term evolution actually was reserved for development of organisms. And that was really a question, how does an organism evolve from a fertilized egg to a fully grown organism, being a tree or a butterfly or a man or a giraffe? And I've been reading a book of Richard Levantine, The Triple Helix. I think he published this in the late 90s. I'm not sure, but an excellent book. And he was an evolutionary and developmental biologist of high caliber, well-known, reputed man, good scientist, definitely. And he makes, in the introducing chapters, he makes exactly aware about this most profound and deep question you were just asking before: what gives form? What is the plan? How is this organized? And he said that we stopped asking that question once we became aware that there are genes. That's now 1860, 70, Mendel. That there are gene mutations. Now we are in the early 20s. That the genetic code is on the DNA. Now we are in the late 50s and 60s, and in the 70s, gene regulation occurred, and of course this took a lot of attention, and it's fantastic how much we discovered, but we lost the question. We thought that genes giving answers to all the questions we had in the past, and that's wrong. We have in our body roughly 23,600 genes, and they allow the production in combination of two genes, sometimes the production of more than 30,000 enzymes. And of these 30,000 enzymes, about 10,000 are active in every cell 24 hours, the whole day. And how this is organized, I think it's almost pretentious to claim that one would understand how this is possible. It's pretentious, but I'm not being pretentious. I just cannot imagine that all the molecules that are on their way to be part of the cell organizations, that they randomly bang correctly into each other to deliver the required products. I believe that there is an organizing principle within this that helps bringing the right material together, and these are the fields.
Right.
So I'm just going a little bit into the direction of answering your question, but only humbly and a little bit, that I believe looking at molecules only, this will never deliver a satisfying answer. But adding what is really there, electrostatic and electrodynamic fields, adding this to molecules, we can feel it a little bit better. I would like to make a simple analogy. Galileo was looking at the planets and realized that they move around the sun. So there's a movement, and the term solar system arised. It works only because there's, apart from the material, the planets, the matter, and the sun, the billions of tons of matter, it only works because there is also gravitational force. And together, the field, the gravitational field, together with the matter of the planets, together, they play a game which we call the solar system or the solar system dynamics. Similarly, but millionfold more complex, is what is going on within a cell. There are molecules, they are charged, this produces fields, and the fields feed back on the molecules, and together this creates a game, cell dynamics. And if you are looking for the plan or the organizing principle, we first of all, that's what I believe, have to look at this, allow me to call it a pair, matter and field. We first have to see how that pair plays and dances with each other. And I think then we might be a step closer to this biggest enigma of life.
No, that's great. As you step through the chronology and how we came to look at genes and then assume that their regulation and action governs everything. What do you notice? I mean, it sounds like you've read papers from scientists from the 30s and 40s and maybe even further back. What do you notice? How have scientists changed themselves? Have they become afraid to experiment and think their ideas? Do you see that, for instance, neo-Darwinism and the current thought is channeling people and making them stay inside narrow courses and not go out and think outside of the box or think these things? I mean, what do you see as happening to science itself over the past 100 years or so?
Yeah, that's a very, very interesting question, and I try to be humble in my answer, because it's pretentious to claim I know what happened over the last 100 years. But I was listening to a TED talk by a philosopher of science, and he said the reason, it's very dramatic. He says that our societies are in a decline, and he says one of the reasons is that the crazy figures that may occur or appear in universities, these crazy figures are not held but thrown out. So the out-of-the-box thinking people, they are not held like a jewel, but they are kicked out. So it is as if there is a kind, let me put it like that, certain thoughts and dogmas and theories, they almost become religious. Like, this is what we look at, this is what it is. Either you do it or you do not. End. And so, for example, when I was studying biology, and I highly appreciate my teachers, and I'm greatly thankful to them, and I really mean what I say. I really mean that. But there were never real discussions about, could it all be different than how we explain it to each other? And if so, how would we need to look at life to find out whether it's different? This type of question never occurred. And then it's also, how to say, very quickly, you are called a vitalist, someone who believes in a kind of a life force. And the moment you are called a vitalist, you are already kind of, how do you say that, cast away. You are already out of
ostracized.
Yeah. It's and what I'm talking about with these fields, this is not vitalism in a sense of there is a life force, because I wouldn't know what that life force is. I can believe it for myself if I want to, but I wouldn't know what it is within the cell. But when I say there are charges and energies that we can measure that really exist, and we don't call them life forces, we simply call them electrostatic fields and electrodynamic fields. They can be measured. They are thousandfold demonstrated. And why people are not looking at this, I do not know. There are some simple sentences like never give up a running horse. So if you get a chance as a professor with a particular theory which you kind of mastered the theory and the experiments that go along with it, why should you change? Because the moment you change, you become a student again. So you have to maintain.
That's a good thing.
Yeah, you understand. You have to maintain kind of your position, and to be a professor today, that's a hard job. It's a hard job.
Yeah.
Sometimes I almost have pity for them because there's so much they need to do, and then you cannot. I think you cannot handle the situation that because when you think in fields, it's another way of thinking. You even need to learn to think differently than from conventional thinking, where we are often asking for a receptor that responds when there is a contact with a chemical, and then there is a chemical cascade that goes down to the DNA, and this leads to a gene expression, and the enzyme that is produced then produces a molecule which is the response to the chemical signal that came in. It's all beautiful. I don't say this is stupid, not at all.
Right.
But also a particular way of thinking. But when you start to think in waves that are non-local, that are extremely fast, and can also contribute to the organizing of a cell, then you need to learn to think differently and to ask differently. And that's an effort. So that's one thing that happened to science. Another thing could be that, I have to say it, the pharmaceutical industry is interested in molecules and not in fields, because molecules you can sell, and science also needs to be financed. So it is possible that that's part of the answer.
Yeah.
Why we still do not look at the fields of the cell. And there might be other reasons. It's technically not so easy to in a femtoliter of volume, to look for these fields. It's not easy. It's a difficult task.
Yeah, that's true. It's true. Imagine what the cells of our body are doing inside our body. How many ways they're communicating.
Yeah. Yeah, it's amazing. It's amazing. But look, even when we communicate, I mean, in an analogous way, when we sit in front of each other, how many languages are actually used simultaneously? We look at the body gestures, we look at the skin color, we look at the height of the voice, high, deep, low, breath.
Pitch, yeah.
Yeah, all these things, and then we also listen a little bit what the other one says. But
it's funny in physics. There's dark matter and dark energy, and they contribute, or they comprise the vast majority of supposedly what's going on. And then in let's say in biology, it seems like again there's just a gigantic dark component that we don't see, that probably comprises most of what's going on. Like you talked about the electric fields, for instance. And then in, human communication, like you just said, the words are one part of it, but the dark part of it is the pitch, the tone, the visual cues, the body language. I mean, everything. It's funny. It just seems like that happens at every level. There's this huge component that's unseen to us normally, that's there.
Yeah. And I think when we look also with the open eyes of a child into the world, when we don't fix what we perceive by our thoughts, when our thoughts do not become a filter for what we allow the world to be, but when we are just open-minded with open senses, we sometimes get that feeling that there is more going on the planet. And I just wish to encourage everybody who has that feeling, first of all, to take oneself serious, but also to be a bit critical and skeptical, not just believing anything, but with curiosity, discovering who we are and what the world around us consists in total.
Well, Daniel, very good. I really appreciate you coming. If you want people to contact you, what is a way they can do that? Do you want to give email, or do you just want to have this interview out there so that people can think about what you said? And what do you prefer?
You're asking me how people can contact me?
Yeah, if you want them to do so, it's up to you.
I mean, It's not easy. It's not difficult to find me. You Google my name and cells, and you come to ResearchGate, and there is a possibility to get in contact with me, or you just give them my email address. They can contact me. It's no problem.
Okay. All right. Well, very good. I appreciate you coming on the podcast.
Wonderful. So thank you very much for the questions, your interest, and giving me an opportunity to explain a little bit how I see the world and what I would love to see in the future going on in biology.
Very good. You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.